Okay, so we're moving on to the last speaker of this 50-minute session, and it's Kazuyuki Hoshijima, and he's going to tell us about homologous recombination. Yes. Thank you.
Thank you for organizers for providing me opportunity to talk about recent progress on the precise editing of zebrafish genome. Okay. So double-strand break introduced by the TRN or CRISPR typically induce indel mutation by homologous end joining. However, in the presence of the donor DNA, it can promote homologous recombination between the donor DNA and the host genome. So importantly, the long stage of the donor DNA can replace the small sequence and integrate the, sorry, it's hard. Okay, so can replace a small sequence and integrate a long sequence simultaneously. So with this procedure, I cannot show the mouse. Where's the mouse? Come on. No, it doesn't. It's okay. Go ahead. With this procedure, we could create a variety of the modification as described here. Okay. In my talk, I would like to present several points to do for the successful recombination to complete these modifications. Okay. First of all, the double-strand break are absolutely necessary to initiate homologous recombination. In other words, it is critical to induce double-strand break with high frequency for successful recombination. So I will show an example briefly. Okay, so I can't touch anything. Okay, so this golden B1 mutation exhibits the pigmentless phenotype because of the premature stop codon marked in red. Okay, so we could repair this mutation by homologous recombination with the wild-type donor. However, it only occurs in the presence of the double-strand break nearby. Okay. This recombination also confirmed by genomic PCR with primer that is specific for the recombinant allele, and also compared by the control allele that amplify from all allele. As shown here, among the donor-injected embryos, come on. Where's the mouse? Okay, it's okay. So it's calm. Oh, good. Okay. So among the donor-injected embryos, these 4 embryos didn't show any recombinant product because of there's no double-strand break. However, these 4 embryos has a recombinant product because it has a double-strand break. So thus, the homologous recombination is totally dependent on the induction of the double-strand break. As second, it's okay. We found circular plasmid are more effective than the linear molecule as donor in our experiment. So we suspect the injected linear donor are not stable in the zebrafish. So we try to stabilize the donor DNA, linear donor DNA with ICE1. The ICE1 is a strong binding affinity, one of the digested end. So therefore, we designed the donor DNA where the ICE1 could bind the end of the insert after digestion. So we expected the bound ICE1 could protect the donor DNA from the degradation or concatenation in the zebrafish embryos. To test these ideas, we prepared a donor plasmid that integrate GFP reporter into exon 1 of KCNH6A gene. This donor had the ICE1 recognition site, both end of the insert. So we have injected this plasmid with tRNA RNA to cleave the downstream intron and analyze with these recombinant specific primers. As shown here, the injection of the donor plasmid with tRNA, we could see the recombination in 6 independent embryos. However, by addition of the ICE1, the amplification becomes stronger, indicating the ICE1 enhanced recombination efficiencies. We try to measure this enhancement more quantitatively by qPCR. Okay, again, this donor DNA was injected with tRNA and ICE1, and then the individual embryo analyzed by qPCR with these recombinant specific primer and normalized with control primers. As shown here, the recombination efficiency is increased dependent on the ICE1 additions. We also pre-digested the donor DNA with ICE1 prior to injections. Still, we could see the enhancement by recombination efficiency by ICE1 addition. So this ICE1 effect also effective in the germline. So among the 14 F-cell found where donor was being injected, so we could identify the 2 of them that inheriting the precisely targeted allele. Because the target gene, the KCN6A, encoding cardiac potassium channel, the homozygote had a heart defect with cardiac edema and expressing the GFE in the heart. So thus, we can say the ICE1 digested the linear donor are effective for the recombinations. Okay. Next, not next. So likely that any method that stabilizes a linear donor molecule would be effective for the recombination. So next, we have asked how to detect the genome modification that are silent. So we try to co-integrate the reporter gene with the desired editing change. So as test case, so we prepare the donor DNA in which golden B1 mutation has converted to the wild type sequence, and range-specific venous reporter has been inserted into the downstream intron. With this donor, so actually we could recover the fully pigmented embryos that expressing the venous in the lens. So I don't show the data, but we have confirmed this embryo has a copy of the donor sequence accurately integrated into the golden locus. So next, we adapted this linked reporter system to generate conditional mutant. Okay. This donor DNA has a pair of the LOXP sequence flanking the exon 6 of the KCNX6 gene. And again, the range-specific venous reporter was inserted into the downstream intron. The recombination of this reporter was stimulated by the double-strand break in the downstream intron. Okay. Among the 43 F-cell founder, we could isolate 6 founder that bred the venous plus F1 embryos. And subsequently, we could identify that 2 of them had precisely targeted allele. Next, we excite the target exon from the KCN6 gene LOXP allele by quick recombinase. The first, all embryos from this heterozygote of this allele looks normal, and then most of them express the venous in the lens. However, by injection of the Cre messenger RNA, this venous expression no longer detectable, and the homozygote show the heart defect with cardiac edema. So these results indicate we could make a conditional mutant mediated by Cre in zebrafish now. So the third point, so we emphasize the co-integration of reporter gene allowed for the easy recovery of edited allele. So we further asked, as fourth point, we have asked if the donor DNA must carry the homologous sequence very close to the double-strand break site. To address the question, we prepared a series of donors with various deletions. The first one is the same one that I used in the previous slide. It has a complete homologous sequence on the right side of the cleavage site. However, on the left side, it has only 16 base homology before interrupting the non-homologous sequence. The second and the third donor that has a 150 base deletion on the left side on both sides. So the recombination efficiency of these donors was analyzed by the genomic PCR with these primer for the on the right side and these primer on the left side. Okay. With the with first donor, so we could see the recombination in eight independent embryos on the left side and the right side. So, and next with the second and the third donor, still we could see the recombination on both sides. And then the recombination efficiency not look like change so much from the first one, although the some embryo, a few embryo less efficient in the donor 3. So it would be interesting if you can see the recombination efficiency on the left side, on the right side. It's similar each other in each embryos with any construct. So we try to further delete the homologous sequence around the cut site. This fourth construct has about 500 base deletion on the both sides. Okay. So surprisingly, so even this big deletion construct, still we could see the recombination in some embryos. Although the other embryo is completely inhibited, the recombination. So these results suggest the small deletion around the cleavage site are tolerate for the recombination. But the big deletion, big deletion made it unstable. The final point I have addressed is that our sequence mismatches between donor and the host sequence tolerate for the recombination. So we found that this K6 locus has a severe polymorphism in wild-type AB strains. So each dot on the haplotype A, haplotype B is SNPs each other's. So we prepare the donor plasmid from each haplotype. Consequently, the left arm has about 2% difference each other, and the right arm has a 1.5% difference each other's. So we have analyzed the recombination efficiency of each donor in the embryo with matched genotype or mismatched genotype. So first of all, as shown here, in the embryo with matched genotype, both donor A and B has induced recombination efficiently. However, in the mismatched genotype, so for example, the donor A in the host BB embryos, the recombination efficiency is severely reduced. Similarly, in the with donor B, the recombination efficiency was reduced in the mismatched genotype embryos, AAs. So these results suggest the identity between the host and the donor's sequence affect the recombination efficiency, and also said that to achieve efficient recombination, you should prepare the donor homologous donor arm that would be highly homologous to your host target genome. Conclusion. It's repeated, I said. It's first, the double-strand break is very important, or it's critical. And then digestion of the ICE1 would help for the recombination. So co-integration of the link to reporter make it easy to identify the silent gene modification. Fourth, the small deletion will be allowed. And then finally, 2% mismatch between the donor and the host are measurable effect on the homologous recombination efficiencies. Okay, the acknowledgement. So this work was done in the David Grant's lab in the University of Utah with the help of the lab members, especially Mick Jarnick, who significantly contributed to this work. And then also thank for the fish facility and the mutation generation core, and then Christine and Christian for providing the plasmid. Thank you for attention. I have, yeah.